lateral feet, parapodia, and on those parapodia they have bristles that block them. They use for traction and stuff. So that would be a good thing. Because the things about the polypeta you had to know were they're marine worms, right? That was one. You should know that they have many bristles, with what the name polypeta literally means, and that they have parapodia. They have these fleshy side lobes on their body, right? Okay. Let's see here. What else could I ask about? Remember, usually on these lab exams, I'll have a specimen, like I'll have a polypeta in one of the jars here, and then I'll ask a couple, 3 things, usually 3 or 4 things. So the class name, polypeta, I could ask you what that means, the many bristles and the parapodia. What was the big theme with the antlers? I described like a big major body design theme that they had. Yeah, the tamarism. So certainly, either with polypeta or with the oligopeta, I would probably ask you to tell me the term that's used to define the segmented body plan, right? A body plan is made up of 2 segments is called the tamarism, right? That will certainly be asked on one of those analytic stages. I don't know whether it's polypeta or oligopeta, but the segmented worm, the theme was the segmented body plan, right? So that's pretty good for polypeta. There'll probably be a couple stations with earthworms, oligopeta, right? And certainly on one of those stations, it'll be the class name. Tell me the class name for oligopeta, which means few bristles, right? That's meaning oligopeta. And then, so one station of the earthworm will be a whole earthworm, like just a whole specimen. And a couple things I could ask you on this. I could ask you what the name of that swollen region, like, around segment 30 is. What's that called? Yeah, the clitellum. And I could ask you what that's for, right? What's the purpose of the clitellum?
Secreting mucus for reproduction.
Yeah, so it's like secreting a mucus cocoon, right? They kind of go head to head, and then they produce this cocoon around them and lay eggs inside of it. Okay. I could also I could also ask you what the gender of the earthworm is. What is it? It's both. Yeah. So if I ask you gender and it says male, not female, you circle both. Right. Okay. It's a trick question. But it's not a trick question if I tell you. Right. That one is fair. And then I'll also have an earthworm dissection. So in the earthworm dissection, I'll have it cut open and I'll have it pinned out, probably with a dissecting microscope there as well. And so, what do you think? What do you think I would pin? Like, if I had 3 things I was going to pin, like organ A, B, C. What do you think I'm going to do? Crop gizzard and something else, right? Crop gizzard, maybe pharynx, right? Maybe typhlosoal, right? Okay, good. So be prepared for those. I told you that I wasn't going to be able to probably pin them free because I can't keep it fresh and visible for the whole depth. But I could ask you a question about what the internal dividing membranes inside the segments are called, right? What are those called? Septae, right? Septae are these internal dividing segments that are made of mesoderm that divide the body into these 2 compartments. Septae, crop, gizzard, pharynx, pretty much. It'd be hard for me to really ask you about the hearts because I can't really put a piece in the heart. And over the course of an hour, their blood vessels kind of constricted, so the people who got their labs wouldn't really see them, right? So just think about, predicting what I might ask you, things that would be easy to pin and easy to pin. All right? All right, so there's 2 Hollywood students probably, and one dissected and one whole. Any questions from those? What about the leeches? So I'll probably have a leech on there, especially for Halloween. And then class names here at the end. And then what other things could I ask about the leech? Like what was interesting about the leech?
No internal segments.
What's that?
No internal segments.
There's no internal segments. There's also no external PD, right? They don't have any bristles on the outside. So that's why the name PD is not in the perineum. And what do they have though that the other ones don't have? Yeah, they have the sucker, right? Oral sucker and an anal sucker, and those are used for locomotion. They kind of and feeding. They latch onto their. Did anyone watch that Stand By Me?
Yeah.
That's pretty funny. No, you didn't watch that song. Great.
Of course I
No one knows that song. That's all good. The leech would just be one of the ones in the vial. And so I'd ask the class name. I'd ask you what unique features it has, which is the oral and anal sucker. It also has no internal septi or division, and it lacks teeth. That's probably the main questions like that one, right? Sound good? Okay, so I think we're through with this. Again, remember the term metamerism. That was a big theme here, and that just means the segmented body plan. Okay. Okay, and then it would probably be worth your while to not only look through the PowerPoints that I have posted on campus, but also to look through your class. And there you go. Okay, there we go. Yeah, so you should be sure to check out this movie. It's pretty fun. All right, then we went into the mollusks. All right, so the mollusks are a pretty diverse group. I'll have probably a specimen of each one of these classes, right? So you'll have a chitin. Polyplecophorae is a chitin. And the chitin, I think I said last time, it was like I had one of those small dishes and I had one upside down and one right side up, right? And the chitin, Polyplecophora. And the chitin, main questions to ask about that one would be the mantle versus the foot, right? I had like the foot was pretty easy to see in the middle. Here's the chitins here. So if you see something like this, here's the foot, the locomotion structure, and this fringe around the outside is the mantle, the part that secretes the shells. And then it'd be pretty easy to ask about what's located in this groove here. You can usually see it if the specimen's set up right. What's located in the groove between the cnidia, right? The gill. So the cnidia is a good one to ask there. And that's about it for that one. I think Polyplecophora, the class name, certainly the phylum name, Mollusca. And it's fair enough for me to ask kind of the phylum names multiple times. Like I could ask the phylum name on the chitin, and I could ask it again, with the clam or something. So just don't let that trick you up. And then the cnidia, foot, and mantle, those are kind of the main themes on there. Got it? Anything else I could ask about the chitin? I don't think I have any radula handy right now, so I probably won't ask about that. But was just thinking about seeing this radula video. Okay, then I'll probably have a couple clams. I'll probably have like the big clam in the jar that's over there, right? Asking you about the class name Bivalvia. I could ask you about the foot versus the siphon, which are labeled on that jar, right? Make sure you kind of know which one's which. And then could also have a clam shell, one of the shells I have, and ask you about features like the umbo and the orientation. So be prepared for, identifying the umbo, be prepared for identifying maybe the anterior versus posterior adductor muscle or adductor muscle scars on the shell. And what I'm kind of getting at with you on that one is that you should be able to tell which is the right shell, which is the left shell on the clam. And what parts, the umbo is dorsal and the gape is ventral. So if I gave you a shell half, you should know if it's the right half or the left half, basically, right? So if you want to dig around in the two drawers over here and just see if you can check yourself on a couple of clam shells, that might be a good thing to do. So, and I'll probably also have a clam dissected and cut open. And in the clam dissected, the main features that I could probably ask would be the foot is pretty easy, right? It's kind of a firm, fleshy thing in the middle of the foot. And then alongside the foot, the tenidia were those gills. Also asking you about feeding methods of the clam, right? The clam specifically is designed for filter feeding. That's what they specialize in doing. And then the mantle is this kind of gland that goes along the fringe to the outside. Any questions on the clam? So like probably 2 or 3 clam stations. What I'll probably do is 2 stations, one with the dissection and the other station with the clam in the jar and the shell. Asking you about those features. All right? Okay. Okay. And then for gastropods, I can't remember what I have on there. I could put an abalone shell. I could put a couple shells. One thing I've done in the past is I put a couple shells out, like an abalone shell, and then like a nautilus shell. Let's see here. Nautilus shell, maybe a cuttlefish bone, and kind of ask you about these, the class names the shells come from, maybe what the shells are doing, right? So for gastropoda, oh, I know what gastropoda is on there. The slug, right? The banana slug.
I was gonna say.
So the banana slug was important because it was a terrestrial mollusk, terrestrial gastropod. And so the banana slug is a good gastropoda, and it has this opening to its, what's called a lung, but it's really a mantle cavity. And so you should know the name of that little opening that goes to the lung. It's called the new shell. So this is a good thing to know. And that just literally means lung mouth in Latin. But PNEU means lung, or respiratory bag. And stone means mouth. But that's a pretty obvious feature on the side of the slug that it has an opening that it can use to regulate water loss and air coming in and out. Got it? So yeah, the gastropod I'm going to use probably. Okay, questions? Looking good? So that'll be one station. Then I'll probably have a couple stations of squid. And I'll have one that's a whole squid, probably asking you some external features, like the mantle or the foot, the siphon. The siphon is the posterior side of the squid. So if you see, for example, the siphon sticking up in the pan, like this orientation, this is the posterior side facing up in the pan. If you see the pen side of the squid, that's the anterior side, right? So you should know the orientation of the squid. The tippy point of the mantle is dorsal, right? This is the dorsal side, the very tip of the mantle is dorsal. And the tentacles and the head are ventral. So that's certainly a whole squid in the pan. And I'll ask you about the orientation, like if it's siphon side up, posterior side, basically up in the pan. I'll have another squid cut open, sected open. And the main features I probably, well, what do you think I'd ask you on the squid that's cut open? We'll keep some good things to ask. What's that? Maybe. Male or female, sure. Probably not so loud lecture exam. Yeah, lecture exam. So the beak, I've never asked about the beak, that'd be a good one. But the sex, male or female, right, we saw those, that's pretty easy to tell. And then the conidia, right, were pretty obvious. And what's at the base of the conidia? Branchial hearts, right? The branchial hearts, which are these high pressure hearts at the base of the conidia, that's a good one because, you know, it has to do with what Brittany was saying about the open versus closed circulatory system. And then I mentioned that the cecum and stomach were kind of hard to determine, especially if it's a female with covered with egg sac. So I probably don't ask that. But what about like the egg sac? Where's the egg sac located?
Right next to the nose.
Yeah, so the rectum tube, little rectum tube, that the egg sac is located there. So that's a good combo question. I could ask you like the pin is located in the blank, you know, egg sac, and where, you know, or the question might be something like, you know, the egg sac is located at what structure, and it's on the rectum, basically. Okay, so it's probably more so what we look at is like the conidia, branchial hearts, egg sac and rectum. Never asked the beak. That's a good idea. Maybe we don't consider it. Problem with that is I have to kind of pull it apart to get it. But anything else there? So I'm not going to ask you about any of the sex structures, but you certainly will have to know whether it's male or female. That's pretty easy, right? The females have kind of a gelatinous egg mass and those big white glands, those shell glands that are on top, called midmental glands. And then the males had like a seminal vesicle, like milky pom-pom, and then it had like we call it a penis, but it's basically kind of a sperm-producing structure. But all you have to know is male versus female. You don't have to know the names of the parts. Okay? So squid, two couple squids, one whole and one cut up. What else? Okay, and then like I said, maybe cuttlefish bone or the nautilus shell or an abalone shell, you have to kind of know it's certainly what the class name of the organism is. And for the cuttlefish shell and for the nautilus shell, what's the function of those shells other than protection? Buoyancy, right? They help them float. So the cuttlefish bone's real porous, almost like pumice, and it helps cuttlefish kind of maintain a little neutral buoyancy in the water. And the nautilus shell is full of gas, and it's able to use it like a submarine and helps float. Okay. Sound good? Hey, did anyone watch any of this, like the octopus, mind of the octopus thing? Pretty cool. Alright. Okay, then we got into arthropods. So, you know, this would be a lot of arthropods in the test. And so let's see. Trilobites. So there'll be trilobites at the station. So I can ask you about trilobites. And just a few questions though, like subphylum name, Trilobites certainly. I could probably also ask you what the name trilobite means, right? What does the name trilobite literally mean? 3 lobes. And that has to do with the kind of the 3-lobe body where we have the middle lobe and then the 2 lateral lobes, right? It's kind of has these triple lobe body. So trilobita literally means tri, 3, and lobita, lobes. It has to do with this middle lobe and these lateral lobes here. They all have this kind of body plan there. And then I have those 2 fossil trilobites. I think probably a good question there would be about what's the theme that arthropods have for their segmentation. They have metamerism, they have segmentation, but they've taken it a step farther, and it's called what?
Diagnosis.
Tagmosis. So tagmosis is sort of the theme of these, and so you should certainly know what that term means. Probably trilobite would be a good place to do that. And what that means is they have metamerism, they have a segmented body plan, but they have they've specialized segments for function, like heads, thorax, abdomen is kind of the theme. Even though those terms are not used for every group, they should just use head, thorax, and abdomen for all of them. They should provide that. But that's the theme of what tagmosis. So certainly you should know that term tagmosis and what it means. Probably would be a good one to ask that. Okay. Okay. Then we have these. I'll probably have a couple stations with chelicerates. The horseshoe crab is a good one to ask. Certainly because the name Xyphosura is so weird. You know, to ask you the class name for the horseshoe crab. And the other thing about the horseshoe crab that would be good to ask would be about the body tagma are called something different, right? The front plate, or the front kind of the head thorax segment here is called a prosoma, right? That's they of course can't call it a head thorax, they have to call it something else. It's prosoma. And then the posterior segment where the gills are located, the book gills, is the opistosoma. So you should know those terms. That would be a good complicated thing. And then the telson will come up again and again. In this case, the telson is a little tail that helps them flip, break themselves up and orient themselves in the water. The telson is the very last segment. But in the scorpion, the telson was the stinger, right? And in the spider, the telson was the spinnerets that make the web. So that telson can be modified for various functions. So for the horseshoe crab, prosoma, opistosoma, and then the book gills were a good thing to ask about that because that's their respiratory feature. The book gills are located here on the opistosoma. Anything else about horseshoe crab? I think it's good, right? I'll use the little one because they're complete. They have all their parts still there. All right. Sound good? And then I like to have that scorpion, that big emperor scorpion on the desk. And the emperor scorpion is good because I can ask about what the name of the pincer leg is called, right? And in the scorpion, it's not called a cheliped, it's called what? Pedipalp. So the pincers in a scorpion are the pedipalps, which are the second appendage of every chelicerate. The sting is the telson, and these slits on the opistosoma, or the ventral side of what should be called the abdomen, but it's called opistosoma. What are those slits called? Book lungs. So you should know the book lung slits on the scorpion. And I often will have a pin in that unique sensory structure that's on the scorpion. When scorpions have it. What is that called? It's like the antenna of a scorpion. Pectens. Yeah. Pectens is the name of that structure. And it's unique to scorpions. I don't think there's a name on there. Pectens. And it's this little kind of feathery thing located just in front of the lung slits, right? It's like a radar paddle. Okay, and then I would like to put that tarantula exoskeleton on there and ask about that. And the main thing there is the fangs of the tarantula or the spider. What chelicerate feature is that called? fanged or modified what? Chelicerae. So the first appendage of all chelicerates is this feeding structure called a chelicerae. Usually the chelicerae is a biting structure. Here's a sun spider or a camel spider, and they have these like really gnarly biting mouth parts. But the chelicerae can be modified into stabbing and sucking for the tick, or fangs for the spider. In the scorpion, it's hard to see them, but they're like chewing structures. The chelicerae is the first feeding appendage of these organisms, and the pedipalp is the second. So the scorpion's pedipalp are pincers, and the tarantula's chelicerae are the fangs. So that's the kind of the theme of these, is they've modified these structures into different functional things. Okay, so a scorpion will be on there. There'll be a tarantula skeleton on there. The spinnerets are the modified tilson. Anything else about spiders? Right. Oh, also arachnida is the class. So we should know class arachnida. And the class arachnida is in the subphylum Chelicerae. That's the complicated part of the arthropods, is that there's a whole bunch of names, right? There's the phylum Arthropoda, subphylum, this group is the Chelicerae, and then the class is the Arachnida. So the difficult thing is getting those names mixed up. Like if I said this spider is in the subphylum blank, and if you wrote Arachnida, I always have kind of a hard time because, mentally, because I'm like, shoot, you gave me a more specific name than I was asking for, but you should know the subphylum name, right? Just pay attention to that. That's one tricky part of this lab exam, is that if I ask you the subphylum name and you give me a more detailed name, I have a hard time wrong, but it's not totally right either, right? So just kind of think of that. I sometimes do half credit for that. Now, if I said the class name, and then you wrote, rather than writing Arachnida, you wrote Chelicerae or Arthropoda, that's wrong. So it's not worth partial credit. So more detail is always better than less detail. Okay, there'll probably be one station that has a centipede and a millipede added, two different dishes. So I'll ask you to, tell me what the subphylum name is for sure, Myriapoda, at that station. And then I'll ask you to tell me which one is, like dish A would be class Chelicerae, and dish B would be class Myriapoda, those names. And then what else did I ask about these things? What was an interesting thing about the millipedes? What's that? Yeah.
They have fused segmentation.
Yes, they have like alternating fused segments and articulated segments, right? So, and what that does is it makes it look like they have two pairs of legs per segment. They really only have one pair of legs per segment, but because every other segment's fused, it's like the legs are double, right? That's kind of cool. And then what's the unique feature of the centipede? What's their Hall of Fame? Yeah, so they've modified their first pair of walking legs into a venom secreting fang. They're not actually using the fang as a sucking part like the spider does. They're using it to inject venom and subdue their prey, and then they chew up the prey. And then also the very last body legs, last are Circe, right? I think my Circe are broken off myself, so I haven't been able to have those for a while. But certainly the fang is a good one. So the first walking leg is a fang, the last walking legs are Circe. Right? Okay. And I don't know. difference in them, like, these are diet. Yeah. Yeah, so one's a predator, carnivore, and one is herbivore or fungivore. Okay, so then we have the group of crustaceans. So I'll probably have a couple stations with crayfish on them, crawdads. And the crayfish, I could ask you a number of things. Like again, I'll probably have a whole crayfish. And on that one, I could ask you some of the leg names, like on the abdomen, they have those uropods, and they have the swimmerets. And then one unique thing, and certainly somewhere on the crustaceans, I have to ask you this idea that they actually have legs that have two branches, right? They have, any one of their legs can actually have two legs branching off of it. And what's that called? There's a term for biramus, yeah. So biramus is the term that's unique to crustaceans in that they have, every leg has like a possibility of having two branches. And you can see this in the uropod. The uropod is the tail paddle of the crawdad or crayfish, and it has like two paddles there. And then all these swimmerets are double branched, like forks. Those are the best ones to ask that on. And then certainly, I'll ask you gender, male versus female, on the whole crayfish. And I don't know, whatever I pull out, it could be male or female. So you should know the difference. And then the name of the claw of crayfish, because, so you compare that to the scorpion, right? The claw of the crayfish is called what? What's the unique name for the pincer? Jellyped. Yeah, jellyped. The name of the pincing leg is called a jellyped. And so, you should know it. What else for that whole crayfish? Gender, probably the uropod, probably biramus appendages, maybe like, didn't I say that their head and thorax are combined? What's that called? If you combine a head and a thorax? Cephalothorax or carapace. I'd probably accept carapace and cephalothorax as interchangeable. Okay. Then I'll have a crayfish dissection, cut open, and probably, like what would I ask on the cut open crayfish? What's that? What do they have? Gills. Yeah, these are called gills, not the other name. The gills are good. Gills would be a good question. Like, what are these feathery structures? Cardiac stomach's a good one, right? That's that kind of cage stomach with teeth in it. Pyloric stomach, if I could get a good specimen. The cardiac stomach and pyloric stomach are good. Sand vein, right? You guys find sand veins in yours? What was the sand vein? Not a vein at all.
So that's so much.
It's their intestines, right? Full of feces. And then pericardium. I can't usually predict that I can get that exposed. Remember when you were second nose, sometimes the pericardium stuck to the shell. Sometimes it was sitting right there in a little triangle. So maybe, I don't know. But certainly cardiac stomach's a good one. Certainly gills are a good one. Pyloric stomach maybe. Sand vein maybe. Okay, so those are which I want you to know that the sand vein is actually a intestine, right? Although some people were able to find the green gland, I have to destroy the thing too much to get there. So I'm not going to ask about the green gland, although I think it's so don't worry about that. Anything else on the second grade? that's pretty good, right? Right stomach, maybe collar stomach, maybe pericardium, probably gills. Fair enough? Yeah. Good deal? Is helpful? Okay. All right, and then I'll have a cockroach dissected on the test. So with the crustaceans, when I ask you to subfile the name, you can either use pan crustacea or so I'll let you do that. But you certainly need to know class crustacea versus class hexapoda, right? Class hexapoda would be including the insects. So I'll have a cockroach dissected on this test. And what do you think I'd ask of a cockroach? Like what would be, if I dissected it open, what if I had 3 pins to pin into things in a cockroach, what would I think? What's that? Okay, malpighian tubules, probably. And the thing with the malpighian tubules is I can't really put the pin in them, right? It's like they're kind of like these spaghetti. And so if you're like in that zone, where are the malpighian tubules located? Like where would you find them? What's that? Yeah, they're at the junction between the midgut and hindgut. So right where the colon or the hindgut starts is where the malpighian tubules are located. And they're kind of a little like opaque yellow color versus the hindgut's more like this, right? So just note that if I have a pin back there and it's not stuck in the gut, but it's kind of next to the gut, and I kind of insinuate these structures, at the hindgut are called laying. Malpighian tubules would be the good one. Because it's a unique structure to terrestrial arthropods. That's how they did their osmoregulation. And that's one of the first things we're going to cover when we come back, is how those work. Okay, and then what else could I ask besides the malpighian tubules? The Siki, where are they located? So like, yeah, the very, very anterior part of the midgut, where the midgut and the foregut are joined. The Siki are basically where the midgut and the crop connect, right? That'd be a good one. And then crop is a pretty easy one to pin, right? And then what about their respiratory structure? What are those called? Trachea. Yeah, so trachea would be good because they're all over. Trachea are those silvery tubes that are all over the inside of the cockroach. So that's pretty good for a cockroach section, right? That's good. Okay, and then let's see, what else could I have on for the hexapoda? Certainly, I'll have like 4 insect feed out for the seed. So be prepared to use your dichotomous feed to identify 4 different orders of insects. Probably will be like 2 easy ones and 2 harder ones, right? So just be prepared for, one being kind of tricky. Maybe it'll look like a bee, but it's actually a fungus. You have to use your key. Got it? I have a key. Okay, so let's see. I think that was pretty good. Oh, yeah. And then, did we cover these? No. Okay, we'll do these when we get back next week.
Tardigrades.
So anyway, I usually have a lab called Echinoderms and Oddballs. Yeah.
He's gonna give us extra credit.
I don't want to forget about
He's gonna give us
You're
extra
so cute. Okay. If anyone wants to come as a tardigrade, one year someone did come as a tardigrade. Halloween costume. No one's ever come as a dodo. Okay, so any questions about arthropods? What did we say? We said there's going to be trilobite, there's going to be bush crab, there's going to be a scorpion and a tarantula skeleton. Hey,
How do we differentiate trachea versus non-tubules?
the trachea are silvery, and they're like, they basically are full of air. So on a microscope, you'll see them look like silver tubes. Malpighian tubules are look like yellow bugs. You know, it's like it looks like. Well, I'm saying not spaghetti because they're like thinner, like super thin, like angel hair pasta. You know, and but they're kind of yellowy colored. And also the trachea are all over, and the Malpighian tubules are only at junctions behind it. So that's, if I had a pin near the hindgut and it's not in the hindgut, it's going to be Malpighian. Okay. All right. So then we finished out talking about animal cells and tissues, right? And so I'll have probably 3 stations asking you about identifying animal cells. Guaranteed there's going to be a frog small intestine, right? Frog small intestine. Let me pull up that on the just on Google Images. Frog small intestine.
Slide. Good slide. See what I see?
Okay, so this is a great slide because let's go to images.
It's like
Check this out. So this is a good one.
it's like a tomato. You wanna get a messed up tomato slice.
Okay, so there's so many things I could ask about this, right? I mean, it looks like kind of looks like something a nightmare or something. But what tissue type is lining the inside here? So the inside layer here, which is kind of reticulated, it's kind of folded. If I were to zoom in on like this, just this layer, just like a single cell layer of like specific tissue type, what is it? Gotta be more specific than that. That's worth, not even worth half credit. It's epithelium, but there's a specific name for it, right? So yeah, so this is the thing, that's why I'm asking this, is remember these tissues that are lining the inside and outside of organs have a very specific combined name. It's epithelium, but there's usually 3 names that go together, right? If it's a single cell layer, what's it called? Simple. So a single cell layer is simple. So you have to say simple, right? And then what's the cell shape? Columnar. And then what's the tissue type? Epithelium, right? So the correct answer for the inner layer, I'm saying it this way, I'm warning you because it's always disappointing or like I always feel sad marking it wrong when you write columnar. And that's all you write. Because the true name is simple columnar epithelium, right? That's the full name. So make sure you use the full name for that. What is the structure that separates epithelium from other tissues? The what membrane? Basement membrane. You can't see the basement membrane, but you can see where it is. See that little line, that sharp line? It's not the basement membrane, but what it is because there's a sharp contrast between the simple columnar epithelium and this connective tissue that's here. This is like areolar tissue inside of it. Okay, so again, when it's epithelium, you have to give me the full complete name. This is simple columnar epithelium. Don't worry about ciliated or pseudostratified or microvilli or any of that stuff. just know simple columnar epithelium would be the inner lining of the frog's small intestine, right? Right? Okay, what's the outer lining of the frog's small intestine called? Stratified epithelium,
Simple
right?
stratified epithelium.
Sometimes it's called stratified squamous epithelium, but I have told you that we're just gonna be more general and say it's stratified epithelium. But, so stratified means it has multiple cell layers, right, Benny?
So then would that not be simple because it's multiple layers?
Yeah, multiple layers are stratified.
So it's not simple stratified, it's just stratified.
Right, right. Yeah, did I say something?
No,
Okay.
I just making
Okay,
sure.
now look back. Okay, so yeah, stratified is the opposite of simple. Stratified means it has multiple cell layers. The name that is usually applied, there's three names. It's stratified squamous epithelium.
Okay.
But when I described it to you in class, I said that it's kind of deceiving because the outer layers are squamous, but as you get more proximal, they become kind of cuboidal. And so, so I think stratified epithelium is good enough for us. But if you write stratified squamous epithelium, that's okay. But that the true name is stratified epithelium, meaning it has multiple cell layers, right? Okay, and then what's the third tissue type I'd probably ask about on this? Smooth. Smooth what? Okay, again, if you just wrote smooth, that's not, I don't know what you mean, but smooth muscle, right, is what the third tissue type. And the smooth muscle is located just underneath the stratified epithelium on the outside. The smooth muscle is this kind of, it looks like a, strands of hair or whatever. It's like basically that kind of ring around the outside is the smooth muscle. Got it? So again, think about when you write the name of these tissues, the name that has to be as complete as possible. So you'd have to say smooth muscle, not just smooth. You'd have to say stratified epithelium, not just stratified. You'd have to say simple columnar epithelium, not just epithelium. So be very specific on the tissue types. And I'll try to write that in the question, like be super specific. I'm not gonna have like 3 spaces. So we can give you a hint, I'll just have a blank, but you have to write the full complete name, right? Sound good? There also is areolar connective tissue in here, but I probably would ask you those 3. The simple columnar epithelium, stratified epithelium, and smooth muscle. That's the 3 main ones. That's why I love this one, because you got like a triple play, out of this one. Okay. All right. And then the other epithelium that was pretty easy to ask was the kidney tubule, right? Kidney tubule, although I showed you this one, the one that we had was actually more kidney tubules. Let me go ahead and see. Okay, here's a good one. So this is what where's the one like this? Oh, here we go. That's okay. Here, these are better. So these are kind of what you saw for the kidney. It looks something like this. And what is the tissue type here? This is the tubule. This is the nephron of your kidney. Simple cuboidal epithelium, right? Simple cuboidal epithelium was the kidney tubule. And so these are the simple cuboidal cells basically circling around the duct tubules of your kidney. And then of course the basement membrane is lining the outside. I put the kidney on there occasionally, but there's really only a few things I could ask about this, right? Like the name of this tissue type. I could ask you about the basement membrane. But the problem with the kidney is it doesn't have many things to ask. What was the other one that had a bunch of things to ask? Wasn't it like the monkey ear?
Yeah.
Yeah. No one's ever come as a monkey ear for here we go. This is a good one.
Ear history.
That's pretty good. You can, there's quite a bit to see in this. Okay, so in this monkey here, what's this tissue here in the middle? Cartilage. This is cartilage. You don't need to know the specific type of cartilage. This is elastic cartilage. But cartilage is pretty unique looking, right? And then what was, this is not a great slide for this. There's also on the outer lining here, you can't really quite see it here, but on the specimen I have pretty good. There's also a stratified epithelium on the outside, right? The dermis. And then areolar connective tissue, basically connective tissue is connected to the cartilage to the stratified epithelium. And there might be some muscle in here too, but I would try to be specific. But the cartilage is the main one. The stratified epithelium on the outside is good. And yet, it's fair enough if I had the same tissue name in different slides, right? Stratified epithelium might be on the frog small intestine or on the monkey here. What other tissue did I say I was going to ask? Cartilage. Bone? Yeah, I think I said I wasn't going to ask you, probably wasn't going to ask you about the connective tissue because it's hard to differentiate, say, for example, this from smooth muscle. It's hard to do that. Oh, striated muscle, right? Didn't I say I was going to certainly ask you about striated muscle? Striated, smooth will be on the intestine. So you got that one. But the striated muscle is the main one because that's our skeletal muscle, right? The striated muscle, we've seen this when we had the trichinosis on the last lab exam. That was in striated muscle. And so that striated muscle will probably be by itself. It'll be on its own slide. The frog small intestine is where the smooth muscle probably be, right? And so if you see what you think is muscle on a slide by itself, it's going to be striated muscle. And the striations are these kind of cross hatches on there. You can see that by rocking the fine focus back and forth. Just very careful. Got it? And I said I wasn't going to ask the third muscle type. I'm not going to say the name. I know someone's going to write it in the test, but I'm not going to ask the third muscle type. What is it called? Cardiac, right? Okay, so cardiac muscle is not going to be a correct answer for anywhere on the test, right? Now, that being said, at least one person will write it. Maybe they'll write it where it says name, cardiac muscle. But anyway, so just beware, and the reason I'm doing that is because the specimens that we have are kind of hard to tell between cardiac muscle and striated because cardiac muscle is striated. It's just kind of braided in its appearance. So, and only vertebrates have cardiac muscle. So, because this is kind of an invertebrate test with arthropods, etc., we'll stick with just striated muscle, and the smooth muscle will be on the intestines. Okay. And what other tissue might I ask? This was pretty good. Like sometimes if I need a station, I'll throw a neuron slide on there. And on this one, I could ask you what this is. It's a neuron, right? And then I think I said it was fair enough if I asked you some things about the neuron. And that was the sensory receiving structures are called what? Dendrites. And then the signal sending system is called what? An axon. That's fair enough. And then all of the accessory cells, all the little speckles and dots and webs around here are called what? Neuroglia. Neuroglia are basically the nerve support cells. That's fair enough. So, frog small intestine, maybe monkey ear, certainly right? So, plan on 3 or 4 tissue types. Okay, so that's So I, for those of you who up late, I'm still working on grading your lecture exams. I'll have the grades post by Thursday. But we won't go over lecture exams until Tuesday of next week. Right? If you have any questions, you can ask. So for today, what you might want to do is you might want to review your notes, specimens, or whatever. You might want to take a look at your key and make sure your insect key works. I'll pull out what we have over here, the boxes that have like kind of all the orders in one box. You might want to just take a look at those and just kind of run through your key to make sure it's usable. I have some specimens in a little tray in the back I can put up in front here with a microscope, and you can see if you can identify what those orders are in the white box. It might be worthwhile going through the cabinet over here, take a look at, maybe you can find the polychaete worm I'm going to use, and look at that clam in the jar and take a look. You know, I've never done this because I'm not that devious. But like, I know that, like right now I have these labeled A and B, you know, I mean, in years past I've switched those, you know, A and B, just to kind of goof with people. Because I want you to know the actual structures, right? So on this one, A right now is the foot, and B is the siphon. But certainly it would be worth your while kind of taking a look at specimens here. I said that we had clamshells here. Unfortunately, I dropped my clam and broke it. But you know, this has been used in the past for exams, and as well as the inside of the shell. So those are the kinds of things you're going to see. So it might be worthwhile just to grab a shell out of here and see if you can identify, you know, which one's the right and which one's the left half of these shells. I had put this on a test in the past, asking what the subphylum and class, you know, phylum name of these specimens are. These are crustaceans, right? These barnacles are crustaceans. So anyway, just sometimes it's just useful to kind of see what types of specimens I've used in the past on tests. You know, these are the only trilobites I have. So you know, you'll probably want to take a look at those. We have some millipedes in here, but it would behoove you to kind of maybe take a look at those specimens, like the spider skeleton, exoskeleton, because you know they're going to be on the exam. We have in the back, in the buckets in the back, we have some crayfish. If anyone wants to pull a crayfish out, take a look at them, reflect, get your memory going. You're welcome to. Okay, I'm good. So let's take a break. I'll pull these insect boxes out and put them maybe on the side tables here. And if you want to take a look at those, there's a couple insect keys here. You can kind of get some ideas. Most insect keys, like I mentioned, start out with wings, whether they have wings or don't have wings. And then they use wings as kind of like their key features. And any questions? Good? Okay, so it's up to you to use the rest of your lab period wisely. If you want to look at specimens or ask me to see something, I can show it to you. If you have something else you need to do, you can do that. Okay, so we're good. Questions? Good? All right. Take a break here and I'll pull these specimens out.